need information from you to be able to represent you adequately. We write position papers and statements of opinion on various topics, and at the moment a survey is out, a mobility survey. It's called Obstacles in Student Mobility, so go check it out. It takes exactly 100 seconds to fill out. And now I will just briefly present our professional partners.
Try again.
YouTube is. What's the address for?
We also collaborate with various student associations, and at this Congress, 2 of them will be present, IPSF, that you could also meet last night already, so the International Pharmaceutical Students Federation, and EPSA, the European Medical Students Association, will be joining us as well. And here, just briefly, our corporate partners. And as I said in the beginning, I will let you know where you can find more information about EPSA, because I know this was a very short presentation in which I mentioned a lot of projects and activities. So around 2 weeks after this event, the EPSA Projects and Activities webinar will take place. This will be a longer presentation where all of the details about different projects and how you can get involved in them will be presented. So it will be online Tuesday at 8 p.m. So I hope to see many of you there. If not, you can always contact us. You can always approach us. There's 30 EPSA team members here. I believe you already saw how approachable we are last night. And yes, if you have any questions or comments or anything about EPSA, feel free to approach us. Thank you. Just to see our faces again. And that will be it. I will pass the floor back to Diogo to continue with the educational part of the symposium.
Thank you, everyone, for a very nice presentation. It's always a pleasure to hear more about EPSA. So, to start now the symposium lectures, we will start with the topic Introduction to Clinical Trials. Nataša Milošević. She teaches pharmacokinetics as well as general inorganic analytical chemistry at the Department of Pharmacy within the Faculty of Medicine at the University of Novi Sad. Oh, I heard some emotions there. She has a diploma and postdoc degree from the Medical University in Novi Sad, and she is now pursuing 3 years in specialization of clinical pharmacy. So feel free to join us, and warm applause.
Thank you. I'm very excited to be here, and I feel very honored. Nice to see you so many coming from Belgrade. I hope you enjoy Serbia. And I'll try to explain briefly. Sorry, I don't know how to use the microphone. Do you hear me when I speak like this? No?
Yes.
Yes? Yes? Can I speak like this without the mic? Yeah? I'll try to speak even more louder. Okay? Yeah? You cannot hear me? Sorry? Okay, sorry. Okay, I'm really sorry. I didn't know it's an obligation, sorry. Okay. Well, a clinical trial is a scientific research study conducted to screen for, to diagnose, prevent, or treat disease in human volunteers or in patients. So there are a few steps in clinical trials, and every clinical trial starts with a research question. Then, according to that question, design of the study is presented, and patients or volunteers, according to their eligibility criteria, are recruited to the study. If required, there is masking or blinding. Data are collected, and then the results are interpreted. There are 2 types of trials, non-experimental one or observational, which I'll present, and experimental or interventional studies, which are the most important studies when you develop a new drug, a new entity, or when you conduct a randomized clinical trial, which are always about an interventional type of study. The observational study means that the investigator does not intervene and rather simply observes the strength of the relationship between an exposure and disease. So we have a natural setting without any intervention. We only observe how our subjects react to some changes or exposures. Well, the first type of study is case report. When you note something unusual, like a new type of treatment, off-label use of drug, some unusual side effect, some disease association, always note it, record it, and publish it. Here is an example of a case report published by my colleagues for our department. Maya is our PhD student, and this is a very unusual case. What happened while she was interviewing the patients for her PhD thesis, she met a young woman who had an infection, a vaginal infection with urea plasma, and she tried all sorts of antibiotic drugs, and nothing helped. And she was trying some alternatives. So actually her mother-in-law advised her to put a piece of garlic, a garlic clove, in her vaginal cavity. And she said, okay, I've tried everything, I'll try this as well. And she tried, and it worked. The infection was gone, and she went to her gynecologist. She was sent to the clinics of infectious disease, and the infection was gone. She experienced no side effects, and the colleagues of our department noted this case, and they have published it this year. So that's a case report. She observed, she did not intervene. She only observed an unusual case, noted, recorded all the medical record, and published. The observational study where you have a similar unusual cases means that we have a case series. So this is an example of case series which was very important since it was noticed in around 66 patients and very interesting interaction between an antibiotic drug, ciprofloxacin, and warfarin. Patients who were stabilized on warfarin experienced bleeding and even death in one case when they were treated with antibiotic ciprofloxacin. Now you can see a note to pay attention when you give antibiotics from the fluoroquinolone group to patients who are stabilized on warfarin. The cross-sectional study means that we have a snapshot of the current situation. So we only observe how many of our patients or volunteers are exposed and non-exposed to some exposition, some change in the environment, and how many are healthy, how many are suffering some type of disease. So we only do these studies to understand the prevalence of various conditions, various treatments, services, etc. This is our study, published this year. We have introduced in our study 102 male participants with normal weight, 51 of them, and 51 obese, who were enrolled in our study to examine the exposition to plastifiers, to be more exact, to phthalates, which are known as endocrine disrupting chemicals. So they interfere with the normal functioning of the endocrine system. And last year we have a cross-sectional study regarding the exposure to female population to bisphenol A, again environmental pollutant, plastifier, found in plastics and in cosmetics like shampoos as well. So the female population was targeted to us, and we introduced 103 healthy females. In cross-section study, we do not study the causality, so we cannot find a connection between exposure and an outcome, and to say, okay, this is the cause and this is the consequence. We only observe the changes. In our previous study, we have found that men who were exposed to phthalates had increased liver enzymes, transaminase to be more precise, and they have increased body mass index and waist circumference. Here in the study with bisphenol A, we have found that a female population who was exposed to bisphenol A has increased body mass index, waist circumference, waist to hip ratio, etc. Whether there is causality or not, we cannot say. We have only a snapshot of the current situation. We only observe how it is. The cohort study gives more answers. That's a follow-up study where you follow your patients during a period of time. In our cross-sectional study that we have conducted, we collected urine samples after 12 hours of fasting. So it means we had one-time exposure to phthalates. If we want to see the exposure during time, we need to follow our patients or our volunteers during some specific time. Here we can see that we divide the observed groups into cohorts, exposed, exposed and non-exposed, and then we follow during time whether they will have a disease or no. One of the greatest cohort studies conducted in the United States, that's the National Health and Nutrition Examination Survey, which is running for years, where patients are included, actually volunteers are included according to a special design where the countries of the United States are separated into groups, and from each country volunteers are, according to a special design, are introduced and enrolled in study. And here are some of the results of these studies. For example, they followed for 19 years 13,000 people in order to see that 10% of them experienced congestive heart failure. They found that males, who are less educated, physically inactive, cigarette smoking, overweight, with diabetes and hypertension have more chance to have a congestive heart failure. So here is the risk factors, and here are the results of the study. The cohort study can be retrospective, so it means we start the study now at this point, and we found how patients were exposed during time according to their medical record, or what changed in their life according to their medical record, so we look into the past. One of the first study I have ever published is the study of blood loss after cesarean section and after gynecologist surgery, and we followed the medical records of 1,000 pregnant women who underwent cesarean section and 500 women who underwent gynecological surgery in order to see how the change of hemoglobin and hematocrit level before and after surgery resulted in the blood loss during surgery. The case control study is also observational study where we find specific cases, disease, and we compare them with healthy participants who have the same characteristics, the same, for example, gender, age, race, body mass index, etc., to see which of them are most exposed to some factors in comparison to the healthy ones. This is a large case control study to see the factors for myocardial infarction. There are 12,000 cases of patients with myocardial infarction were compared to 14,000 controls, and they found as risk factor body mass index, waist and hip circumference, and waist to hip ratio. Interventional study, which is the most important study for us, is a study tailored to evaluate direct impact of treatment or preventive measures on disease. It is run in 4 phases, phase 1, phase 2, and phase 3, before registration, and after a drug is registered on the market, it starts the long-term post-phase 3 follow-up, which concerns the side effects from long-term use. In phase 1, we need to determine the maximum tolerated dose, and there are several designs how this can be done. like standard, where we observe a group of 3 patients with a minimal dosage. And this is the first dose that we test. If there is no toxicity, we increase the dose. If there is any toxicity, we observe 3 more patients. If there is only 1 toxicity out of 6, we increase the dose. And if there is 2 or more toxicities, we stop and we declare the maximum tolerated dosage. Again, we can see here different type of designs for the phase 1, where we can find and determine the maximum tolerated dose. In this phase, we also determine the pharmacokinetics of the drug, like the maximal concentration of the drug, the time required to receive the maximal concentration, the area under the curve, and other pharmacokinetics parameters. In phase 2, we test the efficacy of the drug, and it is run usually on patients who only have that specific disease without comorbidities. And this is one of the designs where 14 patients enter, and if there is no response, we declare that the drug is not effective and we stop further testing. If there is only 1 response, we add more patients into the treatment, and we hope for our drug to have at least 20% effectiveness, so we can say that we can expect 96% chance of at least 1 success. After phase 2, it starts phase 3, which is the most complicated, usually a multicentric trial, and includes patients who also have comorbidities, not only otherwise healthy patients. It is more complicated, and always we have a control group and experimental group. The control can be historical, concurrent, and the best one, randomized, which is the gold standard. Sometimes we compare our treatment to placebo, if it is statically. The placebo effect is well documented. It's a mental effect when the patient is hoping for improvement of the disease only because it's treated. It can be sugar-rate water. There is no treatment, but still there is mental expectation. If I drink the drug, I'll feel better. And it's documented that it can be up to 30% improvement due to placebo effect. To eliminate the placebo effect, sometimes, if it is ethically approved, we compare our treatment to placebo. Here is this type of study. Lamotrigine was tested against placebo in treatment of bipolar depression. The historical control is non-randomized when we find the controls from the literature, and in this trial, mycophenolate mofetil in the therapy of nephropathy was compared with patients who were treated with cyclophosphamide. So we had 32 cases tested with mycophenolate mofetil and compared to patients who were previously tested with treated with cyclophosphamide, and these data for the controls were found from the literature. The concurrent control is also non-randomized, and here is the trial where patients with human papillomavirus positive head and neck squamous cell carcinoma were treated with chemotherapy. And why this is not randomized? Because after the chemotherapy was finished, the consilium of doctors decided whether the patient will receive chemoradiation or whether they will receive surgical resection. So the patient did not have equal chance to receive treatment A or B, but the doctor decided what is the best treatment for them to continue. The randomized controlled clinical trial is the gold standard trial to eliminate the bias, to eliminate the expectation for the care providers of the effectiveness of the treatment. And of course, when you include your patient, you need to define very strictly the inclusion criteria and to explain the participants what to expect from the trial. And we worked through this to our workshop today. Please come, I'll wait for you. And if it's necessary to eliminate the bias and to eliminate the physiological influence, blinding or masking is also included, where the care providers and the study participants do not know the type of the treatment, whether do they receive treatment A or B. Every study needs to define the outcomes which are followed during the study. This is the fundamental design of the study. So we recruit the patients. If they are eligible, they need to confirm by signing the consent. How to write a consent, also we will work on a workshop today. If they sign the consent, they are randomized to treatment A or B, where they have equal chances to receive both types of treatments. Sometimes, to be sure that the patient will follow the advices, so our adherence is very high, we have a running period where the patients are first treated to see how is the adherence. If the adherence is satisfactory, if they follow the advices of their care providers, then they are randomized into treatment A or B. The parallel design means that the patients have equal chance to receive treatment A or B. And here is a parallel design where patients were treated, randomized to have imidaclpril or nifedipine in the therapy of hypertension. Superiority trial needs to show that your treatment is better than the gold standard, better than the control. And here is this type of treatment where it is proved that ginger is more effective than vitamin B6 in treatment of nausea in early pregnancy. Non-inferiority trials means that you test your treatment to be similarly effective as the treatment, as the gold standard, but is cheaper, is less toxic, or is less invasive. Here, for example, valsartan alone or in combination with hydrochlorothiazide was similarly effective as amlodipine in the treatment of hypertension, but it was found to be better since it was better tolerated by the patients. Crossover design means that each patient is their own control, so patients are randomized to receive treatment A or B. Afterwards, there is a washout period and the treatment shifts. In this trial, they tested coenzyme Q10 against placebo, and after 12 weeks with coenzyme Q10 or placebo, there was 4-week washout period, and then the patient received the alternative treatment, which was that patient treated previously with coenzyme Q10 now will receive placebo and vice versa. The withdrawal study means that each patient is treated with the same treatment, and after randomization, there is equal chance to continue with the treatment or to stop the treatment. For example, here, patients were treated for chronic diabetic peripheral nephropathy with tapentadol, and after 3-week period, they have equal chance to continue for 12 weeks with the same treatment or to receive placebo. The randomized consent design means that we compare a treatment with another treatment and with placebo. So patients have equal chance to receive treatment A, B, or placebo. Or in this case, equal chance to receive fluoxetine or placebo for the treatment of depression. If you observe here, what we compare nortriptyline versus placebo, fluoxetine versus placebo, and fluoxetine versus nortriptyline, and vice versa. The factorial design means that the patients have equal chance to, after randomization, to receive treatment A, B, A and B together, or none of them. In the trial here presented, patients treated who have hypertension were randomized to receive valsartan, amlodipine, the combination, or none, the placebo. So we, as you see here, the trial compared the effectiveness of amlodipine versus placebo, valsartan versus placebo, valsartan and amlodipine one against another, and amlodipine and valsartan in combination versus placebo and the single treatment only with amlodipine or only with valsartan. The trial that we are conducting currently on our faculty is the MSc treatment for obese with non-alcoholic fatty liver disease, or so-called MSc for Health project. which is supported by the European Commission. This is so-called Horizon 2020 project, Maria Sklodowski-Curie Research and Innovation Staff Exchange. And this trial includes participants from 10 countries, and here we are, the medical faculty of Novi Sad. And Mass for Health enrolled patients with diagnosed non-alcoholic fatty liver disease, since there is no treatment for non-alcoholic fatty liver disease. There are no what is considered to be gold standard for the treatment of this type of disease. And it affects approximately 50% patients with diabetes and 70% obese patients. In this randomized double-blind placebo-controlled clinical trial, here is the design of the complete study. Here is the multi-center randomized double-blind placebo-controlled trial. It's parallel R, where, of course, it's an interventional trial. It's quadruple blind, so the study participants, the care providers, the investigators, and outcome assessors do not know what is the treatment of the patient. It's parallel R, so one arm, patients have equal chance to receive mastica or to receive placebo. Mastica is a natural product of Greece. I say hello to our Greek students. And it is protected by the European Union protected designation of origin. It is a natural resin, Pistacia lentisicus resin, so-called tears of Chios. On the island of Chios, here it is, there is Masticoporia region with 24 villages that produce traditionally this resin called mastica. And our patient had equal chance to receive mastica or to receive placebo. We enrolled according to very strict protocol design. We enrolled 30 patients, 29 have finished our study. We are very eager to see the result of this study, which is still conducted in Italy and in Greece as well, so since it's multi-centered. We followed the primary outcomes, the live score, and secondary outcomes, variety of outcomes. As you can see, liver enzymes improvement, anthropometric characteristic, genetic profile, metabolomic profile of patient, etc. in 6 months frame. Before and after treatment, patients were treated for 6 months with 2.1 grams of mastica or with placebo. And we have recruited, as I said, 30 patients with very strictly defined inclusion criteria and also very strictly defined exclusion criteria. And about the recruitment of patients and what are their rights, maybe some questions that they have, we can talk on workshop as well. And the patients were followed according to very strict protocol. The lab protocol included blood samples and stool samples at 0 time and in 6-month time period. Of course, some questionnaires, which are very strictly defined and translated from English to Serbian for right patients, which included different type of questions regarding medical history, physical activity, life habits, smoking habits, etc. So we wait for our results, and I hope in some other meeting maybe I will have an opportunity to present how this natural product worked on our patients. Thank you for your time, and if you have any questions. So, any questions for me? Okay. Thank you. I enjoyed. Is there any Greek students here from Greece? Have you been to Chios? No. No, it's not. So I hope it will be famous by next time we see you. See you.
your presentation. Remember that we will have the panel discussion and we will have time to have more questions and have a bigger session. So we will now continue with another presentation. So the topic will be balancing ethics, science and regulation, the Dutch model of clinical trials regulation. So I will introduce our speaker. I hope I get the name right. We didn't spell in accent. Joop van Gerven? Yeah? Good. Yes. Studied medicine at Utrecht University. He obtained his master's in 1982 and master's degree in 1984. He subsequently worked in the Department of Internal Medicine at Leiden University Medical Center. In 1990, he completed the PhD thesis on aldose reductase inhibitor treatment in diabetes mellitus. He was trained as neurologist in Erasmus Medical Center, Rotterdam. From 1988 to 1994, he subsequently completed his training as a clinical pharmacologist. Since 1994, he has been the director as CNS research at the Center for Human Drug Research in Leiden, and is responsible for consultancy and research on central nervous system drugs and methods. He is also professor of clinical neuropsychopharmacology at Leiden University and staff neurologist at LUMC. His primary scientific interest is in demonstration of proof of pharmacology for central nervous system active agents during early phases of drug development. He has published around 200 peer-reviewed scientific articles and book chapters, and is a regularly invited speaker and committee member of scientific organizations and conferences on various aspects of this subject. Since 2011, Joop van Gerven has been the clinical pharmacologist, vice chairman, and acting chair of the Central Committee of Research involving human subject, the Dutch Clinical Trial Authority, and the Central Ethics Committee. In 2017, he was appointed chairman of the committee. So, you're warm welcome to our speaker.
Good morning, everybody. If I knew that Diogo was going to read my summary, I would make it shorter, but it's almost as long as my lecture. So I apologize for this. I also apologize for the fact that I'm a second speaker. I was supposed to be the third one, but I guess the second speaker will arrive in a few minutes. So not to keep you waiting any longer, I thought it would be a good idea if I started, but I do introduce some concepts that might have been better explained in a previous section. I'll do my best to sort of fill that gap. Now, I will tell you about the Dutch model. Now, why would you be interested in the Dutch model anyway? We're just one of 35 countries a member of ESPA, so there's nothing very specific or particular about Holland anyway. But the Dutch experience will tell you some of the dilemmas that we have when we try to combine ethics and regulation. And that's why I think it will be interesting as well. Now, it's important to state in advance that I do not want to make the impression that our system is better in any way than any other system. It's just one way that our system evolved. It has some advantages and disadvantages, and in trying to explain to you how they came about, I hope I will make clear to you that what the sort of dilemmas are that you face when you try to regulate clinical trials, which require regulation, as Nataša illustrated quite eloquently. It's a very complex business, and there are many parties involved, so there needs to be some type of regulation, we think now. We didn't always used to think that, as I will show. So So what I will do is sort of guide my presentation along the guidelines, which really only came into force not so very long ago, at least not from my perspective. Only, let's say, in the beginning of this century, we had the first regulations in ethics at a European level. And this, of course, is now sort of making a step forward in harmonization with the clinical trial regulation. Now, this is the bit that I was hoping was covered to some more extent before I start my presentation, but we'll come back to this. And this is really the topic of my presentation. So, how do we balance the needs of patients and sponsors and research and society, I might add? They all have vested interests in trials, so how do we deal with all those interests which are not always matching? And along that line I will explain why we decided to for a slightly different interpretation of the regulations of the rest of Europe. Very important to understand how the Dutch have set up their system is to understand a bit about the way that the Dutch are. There's a very important concept also in our own perception, which is the polder model. You know what a polder is? Actually it's a bit of sea or lake that is made dry. And we used to do this for over 1,000 years. And of course, there's no single person or no single group of people that can actually do this. It requires a very large collaboration of all the people in the environment or in the vicinity of a polder, because you have to pump the water out of the polder into somebody else's land. They also have to be involved, and this led to very complex but efficient democratic structures which are still in existence today, and they are in fact the oldest democratic institutions in the world. We still have the habit in the Netherlands to talk to each other before we actually start doing things. So this consensus is extremely important in Dutch politics and also in Dutch society and in Dutch science. It's actually a very nice book that you can download for free if you're interested in the subject, how the Dutch came sort of to punch above their weight because of this collaboration, this consensus by the Royal Dutch Academy of Sciences. Now this same sort of consensus approach to problems already led to the first formal ethics committee in the Netherlands over 40 years ago, which was formally implemented in 1979. And that was when I more or less started my medical studies, so a long time ago. And it took some time, for reasons I won't go into, before it was formally implemented in Dutch law. And that was one of the first instances where control of clinical trials, so that is ethics approval, was mandatory in law in any country. And mind you, that was before the European Directive, which basically tried to arrange something very similar from a different perspective. And when the European Directive came into place in 2001, we had a problem because we already had a system that started to be quite useful for us, as I will show you. So we had to match the European regulations at that time with the Dutch system. So this is more or less the basics of the system that we set up in the last century under this Medical Research Involving Human Subjects Act 2001. And it basically entailed that there was one supervising ethics committee, the CCMO, of which I'm the chairman, But there were regional or institutional medical ethics review committees, which were also governmental bodies. So they had a formal role, and they were formally accredited by the CCMO, and therefore by government. The idea behind this was that by sort of adopting common standards in terms of composition of the committees and the way that they worked and the way that they made their judgments about trials, we would reach a certain level of harmonization to the extent where the approval of a single ethics committee anywhere in the country would automatically be also accepted by all the other institutions in the Netherlands. So you only needed one single approval from one single ethics committee to start a research trial anywhere, which was of course incredibly useful for multi-center trials. And that was before the European regulations came into place. We also organized a clinical shift here in the, so this is a mandatory course for clinical investigators. So every researcher needed to be aware of this system and be actually certified according to this system before they could do research in humans. And the ethics committees would test for this when they approved the trial. And it was for all medical research, not just for trials. At least all medical research where people had to do things. So not the type of observational trials, but interventional trials. And this is what we still have in the Netherlands. Now, the number of ethics committees since 1999 reduced dramatically from about 100 when we started to 20 now, 21. And this was all voluntarily. Most of the ethics committees decided to stop or to hand over their work to other regional committees. So there was no force, or hardly any force at all, to get this accomplished. But we still believe this is a good development. We need all the many, they need to have a minimal level of experience and expertise, and we also need to maintain control. This is more efficient with a smaller number of committees. The CCMO also reviews the protocols, but only to a limited extent. We looked at whether the documents are complete, whether there's anything missing, but other than that, the entire process is delegated to a single ethics committee, which is at the choice of the investigator, basically. And we do an integrated review. What that entails, I will go into more detail later. But it's an ethical review as well as a product review and a scientific review. It involves all medical scientific research which subjects humans to research activities, which is most of the drug trials, except the observational ones, but that's only 1/3 of the studies that are being performed as medical scientific research. All the ethics committees and the CCMO approve about 1,800 studies in the Netherlands per year. So what we cover is medical scientific research with human participants, that can be healthy or patient, who are subject to specific research activities or changes in behavior. So it does not entail other types of research, and in fact there's no formal regulation for the other studies in the Netherlands. Now this may seem odd to you, or maybe not, but that's actually the case in all the other countries as well. There's no formal regulation for studies in any country. And in fact, the European regulations only pertain to drug studies within this field of medical scientific research that subjects humans to changes in behavior. So non-medical, non-drug studies also fall under no regulations in other countries, but they do in the Netherlands. Still, we don't do any sort of approval for many important types of research, and it's important, I think, to realize that we only look at the tip of the iceberg here. So there's lots of behavioral research in psychology, for instance. You can all imagine this, but these studies are not formally approved by any governmental body, and hardly ever by any formal ethics committee. Increasingly, people do studies in other faculties as well. The legal departments, economic or technical departments, all want to do research about how their sort of activities work out on humans. Population studies, you can do them. There's no formal regulation. Of course, you have to apply with privacy laws, etc. And general law applies to all these studies, but no formal trial regulations. Non-interventional drug trials and observational research that Nataša describes is not formally regulated. Retrospective studies, again, Nataša described these, not formally regulated. Experimental treatments of the type of the vaginal garlic clove is not formally regulated. Biobanking studies with patients that are with materials that are obtained during medical practice. Animal studies, there are recognized animal ethics committees, of course, also in the Netherlands and in other countries. But the other studies are not formally regulated, and we believe that's not appropriate. We believe that there should be some sort of common strategy towards research in humans. It shouldn't really make a difference whether you go as a volunteer to the medical faculty or to the psychology faculty, except for the type of research. You should have the same rights and the same principles apply to those studies as well. So we are actively setting up collaborations and platforms to deal with this. So that's work in progress, very difficult work in progress actually. The CCMO, so that's the Central Ethics Committee in the Netherlands, also deals with other trials than just drug trials. We deal with the Embryo Act, so research on embryos is also part of our responsibility. We deal with medical devices, which are now also part of a European regulation which has major impacts on healthcare. And also for pharmacy, it really has a major impact if you think about different types of routes of administration and application. So that's for a different subject. Now the CCMO is also an independent accredited medical ethics committee. We don't just supervise all the other ethics committees, we also do clinical trials ourselves, and we do the more complicated ones. For instance, the ones that are non-therapeutic in legally incapacitated subjects and minors. They are all judged by the CCMO. Unregistered therapeutic vaccines. This is complicated stuff, and we have the expertise, which is rare in the Netherlands, so we combine it in the CCMO. Cell therapy, gene therapy, RNA, related to gene therapy, of course, xenotransplantation. This is the research in fetuses and embryos. Now the reason to combine these complex trials into one central authority is because these are sensitive topics from a societal and political point of view, but they are also ethically sensitive, and this gives the CCMO sort of a moral superiority, so to speak. I mean, this sounds boastful, but all the other ethics committees need to have at least some respect for the approval of a central body. And if it would be a purely administrative body, CCMO, which doesn't do the same work as the ethics committees, then we would lose our respect from the other ethics committees. So we have, and instead we are now the ethics committee that deals with the most complicated and scientifically challenging issues, which other ethics committees can respect. And this is reflected in the way that the ethics committees are formed in the Netherlands, and particularly also the CCMO. There's, by law, a dedicated group of people, which involves physicians, of course, pediatricians for committees that do research in children, clinical pharmacologists for committees that do research with drugs, same goes for hospital pharmacists, health lawyers, research methodologists, medical ethicists, and the layperson. And I'll come back to the layperson in a minute. And for the CCMO, because we deal with the embryos, we also have embryologists. And we also have nurse scientists, behavioral scientists, and the basic scientists, because of the complexities that we have to deal with as well. Each of these more, let's say, scientific disciplines in this system has to be expert level, because other researchers, of course, expect their protocols to be evaluated by people who know at least as much about them as they do themselves. So all the members of these ethics committees have PhD levels, and they are experienced and active, so they must still actively publish. And the minimum that we expect, and we check regularly, is that they have to have one international publication at least once every year. So this is a minimum level of expertise. And by maintaining this this balanced sort of expertise, we try to come up with the best possible evaluations of protocols, not just from an ethical point of view, but also from a scientific point of view. Now, the layperson, there's been lots of debate whether this should actually be a patient representative, and we believe that's not a good idea. Perhaps we can go into this more during the workshop this afternoon. We believe that a layperson should represent all patients, not just one patient group, because we deal with all types of research, not just for one condition or one disease. But the layperson should be able to represent other people. So we ask them to be mayors or judges or people from the board of large companies, societal companies also. And then in addition, they should have demonstrable affinity with healthcare issues. So, and this is for all the ethics committees required. Now that's because we don't have a formal governmental body that that would deal with elements of clinical trials. So we don't have a separate governmental body that looks at the drugs, or a separate governmental body that looks at the contracts or something. We all have to do that ourselves. And that gives us a position within the field of all other governmental bodies that deal with drugs. And in the Netherlands, that's quite a few. Actually, in most other countries, that's also quite a few. So this is the research. So we are the competent trial authority. We collaborate closely with accredited ethics committees. We also have the health inspectorate, who looks at the performance of trials in the hospitals. We have the Medicines Evaluation Board, CBG, which collaborates closely with EMA for the registration of drugs. And then we have, let's say, more societal healthcare watchdogs, like environmental or adverse effect, pharmacovigilance, and of course the reimbursement, which is taken care of by the Healthcare Institute. And we collaborate closely together, and when a trial enters, let's say, our realm, we closely communicate with the other agencies to see whether this trial serves any purposes within their own requirements. Now, when the Clinical Trial Directive came into place in 2001, the system was basically set up to include formal approval of drug trials. That wasn't the case before 2001, and European countries wanted to have a separate body looking at drug trials, in addition to the ethics. So they set up a competent authority that looked at the trial in a stricter sense, next to the ethics. And this split, basically, trial approval and ethics for drug studies, at least. So the drug registration authorities, in almost all countries, were made responsible also for clinical trial regulations with regards to drugs. And of course we had a different system. In the Netherlands, this was combined. We looked at the protocol and the ethical documents, like the informed consent and the product information also, in the same process. So, because this was obligatory by European law, we decided to assign the tasks of the competent authority to the CCMO, and but to maintain the role of ethics within the accredited ethics committees. And the CCMO now only does an administrative review, and all the contents of both the ethical and the drug registration information is still integrally evaluated by an ethics committee. Now that was in 2001, and well, what this led to is that if a sponsor wanted to do a multinational clinical trial, they had to go to all the different governmental bodies that looked at their product and their protocol. And there were many agencies, so that was a problem for a multi-center trial. And after that, of course, you had even more ethics committees. Look at Italy, for instance. I don't know if there are any representatives from Italy, but let's say earlier in this age, there were more than 1,000 ethics committees in Italy, for instance, and other countries also had lots of different ethics committees. So this was still very inefficient, and at that stage, the Netherlands had a competitive advantage, because by then we only had about 24 of ethics committees, which were all formally harmonized and governmentally approved. So researchers started to look at the Dutch model, because the European directive really caused problems for particularly academic research at that time. And they started to argue that you don't need multiple bodies to show responsibility, but a simple single review system such as the Dutch model. So there were appeals to actually set up something very similar to us, which of course made us proud, as you can understand, even though our own researchers looked at it quite differently. They saw increasing rules and regulations. Now, this will regels, these rules, and much as you can imagine, this is a desperate investigator who wants to get his trial done. And so it wasn't all, let's say, ideal situation in the Netherlands either, but at least it was more efficient than all these different countries with all their ethics committees. So let's look at the advantages of different trial systems in a bit more detail. So we have a regulatory review in the European system by a central agency. Now this is harmonized, it's efficient, and it's integrated with drug registration, so that really is an advantage, typically, particularly from the perspective of a sponsor. These regulatory review institutes are highly professional, and they know the regulations and the guidelines very intimately. So they really are, in that sense, also they represent expertise. But it's an expertise that is limited to that governmental institution. It's not an academic expertise. So there's some dissociation between the expectations and the professional standards for these governmental institutions compared to the academic researchers. It's more linked to the standards of pharmaceutical industries. So they match pretty well with sponsors, but not with academic investigators. The process is quite predictable and reliable, even though it may be quite lengthy at times, because these bureaus, these central bureaus, are really not all that large, and they have limited capacity. But it's administratively uniform. It's usually shared by governments and industry because they adapt accordingly, and because it's based on, let's say, regulations and governmental and formalized, it's also pretty well accepted by society. The Dutch system is much more an integrated review. Scientific, product quality, and ethical are all integrally approved, and that leads to, particularly with sponsors, to questions, why do you deal with the science? I mean, you're supposed to be an ethics committee. Or why do you have questions about the product as an ethics committee? Which ethics committee has questions about a product? And if you're a governmental body, what do you care about ethics? So these types of questions come up with people who are used to this European regulatory review process. At the same time, academics like our approach, and some sponsors who are still searching for the best possible clinical trial, like the fact that we involve top-level specialists, at least within a Dutch perspective. And of course, these specialists also know the local situation, because they are, these accredited ethics committees usually are affiliated with the major centers where the principal investigator for a central trial is located, so they know that local situation very well. One important thing, and I will go into that in the last part of my talk, is that we can do proportional evaluations much more readily than the predictable process of a central regulatory body. And we can look at practicality. All of the ethics committee members, including those of my own committee, are practicing physicians, they are practicing scientists. And so we can be practical also about our approval process. And we can try to disseminate our views among our own colleagues in other sort of groups where we meet them. And because we are all scientists and also part of the scientific community, we can share the guidelines. The development of guidelines can be a shared process. We can distribute knowledge, and the decisions and the explanations for these can also be shared. So this really improves the acceptance of the medical and scientific community of the ethics committee process. Now this afternoon I will go more into detail into what exactly you look at when you do an ethical judgment. Now one important thing is that we look at the autonomy of the patient. So the patient has the right to refuse or choose their treatment. And that means that we look at the information that the patient gets, the recruitment process, how the expectations are given, what type of remuneration they get, and whether there's an independent physician that doesn't try to coerce them into the study because it would be the only treatment option that they have. That type of language we don't accept. The second medical ethical concept is beneficence. So you have to act in the patient's best interest. So that means that we look at the burden of a study, we look at the competence of the investigator and the quality of the research facility and the process, and look at whether there might actually be clinical benefit for the treatment also. Non-maleficence, another important concept. First, do no harm. So that means we also look at whether the patient gets the best possible treatment options, which for placebo can sometimes be a source of debate, but then it helps that we are also practical. We look at insurance, we look at whether there's a safety board and how they work. Justice, fair and equal distribution of scarce health resources. So we want to perform studies in a population that can also have access to the new medicines. And that there can be open label extension trials. We always ask for them if it is possible. Again, practicality limits this to some extent. Dignity is an important concept, just to make them complete, and truthfulness. Truthfulness is particularly important, so we look at the financial arrangements, we look at the contracts, we look at the publication clauses, termination clauses, and we look at the scientific and methodological context of the trial very much. Now these are scientific arguments that you think may not play a role in an ethical debate, but in fact they do. We believe that poor medical research is unethical, and that if you do a trial in patients, it should be of the utmost and the highest scientific standard, and the best and the most should be learned from it, with the least questions remaining. Now this proportionality, and this is almost the final part of my talk, is actually quite useful and important, and it's something that in European regulation is difficult to maintain. We are a clinical trial authority, so we look at clinical research. But we do that in addition to authorities that deal with registration of medication and separate authorities that deal with the healthcare remuneration issues. Primarily, we can make a different level of assessment and apply different quality standards, perhaps even, I would say lower quality standards, but more fit for purpose quality standards to a research project than we would, than the Medicines Evaluation Board would for registration of a trial for market approval, or a healthcare institute would for remuneration in a healthcare package. There are different types of standards which can apply. So we look at scientific rationale, knowing that it is still an experimental treatment. And that means that the product information has to be balanced in that particular context. It's quite different if you give a drug in a healthcare system to thousands of users than if you give them under very controlled trial conditions to perhaps 10 or 20 with close supervision. You can be more lenient with regards to product quality and manufacturing process in a clinical trial setting than you are in a healthcare setting. Patient protection, well, it's also different if you are in a position to very carefully inform a patient with extensive informed consent processes than when you just have to rely on a package insert to communicate with your patient. And the research outcome is of course different. In addition to sort of the personal benefit that a patient may gain, there's also a more general scientific benefit. And almost by definition, research is not performed for the benefit of an individual, but the benefit of the group and society. So that all are different types of evaluation levels that we apply if you compare to other bodies, and I will list them in a second. So that means that our committee members also have to be different, and they have to involve experts as well as laypersons. The drug registration authorities, like the EMA, or the Medicines Evaluation Board in the Netherlands, they look at controlled clinical trials and look at clinically relevant endpoints. And they look at GMP product quality with the perspective of of healthcare release. In the registration of the dossier, they look for statistical significance in these endpoints and whether there are any worrying safety symptoms, which is quite different from what you would do in an experimental study. Now, if you assign these tasks for clinical research to these bodies, you will get sort of tension between what is acceptable for clinical trial. And we get these arguments very often in the European setting. So a European regulatory body for a clinical trial in another country will often ask questions about the product quality that are not appropriate, in our view, for this particular early stage of development, because it's all controlled in a very well sort of organized setting. It's not a manufacturing process for healthcare release. And these types of questions dominate our discussions quite a bit. Now, the European system recognized these types of problems in 2014, and after lots of debate and arguments in the years before that, they implemented a new regulation, which, different from the directive of 2001, now had to be implemented in full in every European country that is part of the European community. And basically it says that there has to be a part 1 in every country, and one country can do the assessment with input from the other participating countries of, let's say, the trial in the strictest sense. So the protocol, the product information, that type of the labeling, the investigator's brochure. And then you have part 2, which has to be evaluated in parallel, and that's more the ethical or national information. So it's informed consent, it's insurance, it's remuneration, compensation, contracts, and local storage solutions provided for samples. Now, this is a system that runs in parallel, and it has very straightforward and fixed timelines. And you won't be surprised to see that we have combined it in our own system, but these timelines really are challenging for us, I must admit. So, and also the expertise that is expected in this system is quite challenging. So we have to remember that in our system the ethics committee membership is expert, but it's voluntary because they're active scientists. It's built on a limited administrative burden, and this really increases in European regulation. There is lots of fixed timelines, and that's not ideal for the Dutch call-ring process, as you might imagine, which was a weak point to start with if you look at timelines only, but for consensus reaching it was actually quite good. And we have to deal with all these European requirements, complicated administrative systems, limited space for proportional interpretation. There's actually no difference between drug registration requirements and research requirements to the extent that we used to apply. And quite frankly, we don't know as much about the European regulations as the European regulatory agencies do, because we have to deal with these experts that we have to train. So we are looking for specialization and support, but it is really a challenge to keep all this set up. So I hope I've provided you in this overview with an impression of why regulations came into place, and they're mainly sort of guided by the support of drug development in Europe, and they're very much directed in that way. So they are meant to harmonize procedures and tailored to the needs of pharmaceutical industries. We believe that the patient needs are still limited, but that's something that we have to implement on a national level, but there's certainly also considerations for transparency in the European regulation. Academic trials still, I think, have difficulties matching all these expectations. but they are largely exempt. At least the majority of in numbers are exempt from the regulations, because the low interventional trials and non-drug trials don't fall under the regulations. And there is an improved integration of regulation and ethics in the European system, as well by the parallel processing of the scientific and product information and the environmental information. And the Dutch still sort of try to fight the incoming water in their folders by sticking to the integration of ethics, science and regulation. So thank you very much. I know that there are, I see questions. There's also a panel discussion at the end, but yes, please. Yes. I haven't talked about the GDPR, so that's the that's sort of the elephant in the room. Several things. So the question, I don't know if anybody or everybody could understand. The question was, how do we deal with the big data and the privacy laws, so the GDPR? Well, actually, there is still debate about this, and in the Netherlands, you have to remember that much of this, if not the majority, is not sort of organized by the European regulations, because they're not drug trials, they're usually observational studies. And let's say all the debate that has now been activated by the implementation of the GDPR also was at a more rudimentary phase going on before the GDPR. And in fact, the GDPR doesn't change all that much if you look at it into detail, except that it highlights old problems that have always been around and that now suddenly seem to be important. They were always important, and the GDPR doesn't regulate them to any sort of important extent. Now, what we as ethics committees do is we look very much at a proportional fit-for-purpose approach, and we try to inform our patients better. So we have a standard patient-informed consent template, which has to be applied in the Netherlands, and that has a section on the data protection and how biological materials are handled. And that has expanded from 2 lines to about 3/4 of the page. But it is still, it basically defines all types of disclaimers. So patients should be aware that if they provide their personal information and their materials in the context of clinical research, they lose some of the control over it, whatever you want to do about it. It's inevitable that there is some loss of control, and people have to understand that they do that. And that's the reason for them not to participate. They should know beforehand rather than be confronted at the end. I could spend an hour talking about the GDPR, which I won't. Thanks for the question. Okay. Well, thanks again.
Okay, we will proceed now with the last lecture of the first panel. We will, after this, proceed to coffee break. But before we proceed to the coffee break after this lecture, please remain seated because I have some announcements to make. Okay? Green motion comes. Okay, nice. Okay, so next speaker is Nenad Miljković. He's a clinical pharmacist from Belgrade, Serbia. and is currently one of the European Association of Hospital Pharmacists, Director of Professional Development. Nenad also holds a master's double degree in European Public Health and is one of the research working group leaders within European Cooperation in Science and Technology Action, European Medicines Storage Research Network. Pay attention because it will be a very interesting lecture for sure. So round of applause for our next speaker.
Hello to all. I apologize that I'm a bit late, but it's very difficult when you work in a hospital and your general manager wants to see you and accept a full report in the morning. It's very difficult to be here on time. So I would like to thank the EPSA on behalf of European Association of Hospital Pharmacists and our president, Peter Horak, for inviting me here. It's always a pleasure to be here. I was in Barlow this spring, and it was a great experience. So we really need fresh blood, and I hope that some of you will follow the path of hospital pharmacy. And today, we agreed in the EAHP, and I informed EPSA that we will be talking about clinical trial regulation. Unfortunately, I was not aware what kind of lectures did you have in the morning. So if you think it's boring, if you've heard that already, just raise your hand, interrupt me, or ask any questions. But today, basically, this lecture is a teaser about the clinical trial regulation and directive. So I work as a hospital clinical pharmacist, half hour away from this area in orthopedic hospital. I also volunteer at European Association of Hospital Pharmacists. So I have nothing to declare. So today we will talk about a bit of historical data, about clinical trial directive and the difference compared to the trial regulation. Then we will try to discuss the goal of this regulation, what will happen when this regulation becomes applicable, then what's left for the member states concerned, and finally, what are the key benefits of this regulation. So if we go into the past, I think the dates are a bit different compared to the presentation before mine, but I've used sources of EAHP. So since May 2004, clinical trial directive was the main legislation tool under which clinical trials are governed in the EU. It was basically the aim of this directive was to harmonize and to centralize the administrative procedure among the member countries. However, it was completely the opposite. So many member countries said that this directive imposed a lot of administrative burden. It was not as efficient as they thought, and in 2011 there was a review of this legislation tool, and they said basically that the directive was interpreted in a disharmonized manner completely. So as a consequence, in 2014, clinical trial regulation was adopted, and this regulation will be active and it comes into application in 2019. This is very interesting for many pharmacists working in the CRO, for many hospital pharmacists, and for many students. And I know that many of you would like to work in CRO organizations, so that's why we decided to present this. So the main difference, if you look and compare directive and regulation, you will see the directive was the goal. And obviously many countries interpreted this goal differently. And the regulation is a binding document. It will have very strict timelines, as mentioned before, and it will apply entirely across the EU. So basically that's the difference, the first one I wanted to present. And then if you look at, I'm not that tall as the Dutch people, so I will have to, yeah. So if you look at the submission process, if you look at the directive, you will see it included multiple submissions, which is something that will be avoided completely when it comes to regulation. And then the collaboration and the review process was completely decentralized. It imposed a lot of burden on each member state that wanted to conduct the same clinical trial. The access to information was also very limited. That will change. If you look at the regulation, it will basically be harmonized approach across the EU via single application process. Then it will introduce a platform which I will present in the next slide. The name of the platform is Single EU Portal and Database for Applications. It will facilitate collaboration between the member state, and the information that will be available to public will be far more comprehensive and basically everything except for personal protection data, which will not be available, of course, and commercially confidential information. Overall, the purpose of the regulation is to protect the patients and the trial subjects, to simplify the application process, and to increase transparency. When I say simplify a clinical trial application process, I particularly mean about the trials which are multi-country trials. So I think it will be less of a headache to those organizations that would like to conduct a clinical trial in 6 countries. The way how they will apply it will be much more easy. The regulation will apply 6 months after the portal is fully functional. I will explain that later on. And the scope of the regulation compared to the directive is not changed. So it doesn't include non-interventional trials. Now a boring slide, but that's the only way we can do this. It's only 2 or 3 like this. So it will increase efficiency through harmonization of the application process. It will enhance safety and efficacy of medicines. It will be, as I mentioned, be applicable only to medicinal products within the interventional clinical trials, not non-interventional clinical trials. It will streamline the process of application for a clinical trial. It will remove the application. We have those a lot. And it will introduce a lighter regulatory regime for trials conducted with medicines that are not considered, medicines that are already authorized with lower risk. Then it will simplify reporting requirements, recognize co-sponsorship, because sometimes you have 2 organizations, 2 entities that would like to conduct a clinical trial. It will introduce the concept of a single decision on a clinical trial instead of separate approvals. And finally, it will reduce the administrative burden. As you will see, you will have a member state concerned and reporting or a reference member state which will be responsible for the phase 1. So this is infographic. I like this because that's the essence of this whole lecture. So in addition to what I said, the transitional document will be put in place. So 6 months after the full completion is confirmed of the portal and the database, the year 1 will start and the Article 98 will be applicable. And during this first year, we will be able to apply both directive and the regulation. In the second and the third year, only the regulation will be adopted, and finally, after that, the regulation will be the only legal instrument to be followed. There will be derogation for all clinical trials which were set up under the old legislation. After 3 years, as I said, of the transition period, everything will have to be carried according to the regulation. We'll see how it will go on. And finally, Article 80 and Article 81 gives the EMA, European Medicines Agency, the responsibility finally to organize, develop and lead with a single EU portal database where everything will be basically sent and application will be created via this portal. This is done after discussion with various stakeholders, so it's not a one-night decision. It's a very thoroughly elaborated decision. And in June 2018, if I'm not mistaken, the portal and the database are considered to be functional. There was an auditable version tested, and hopefully in 2019 we will have this regulation function. Again, a little bit of tables. I think we mentioned this, but what I wanted to say, so now we will have single e-submission portal, and we will have joint assessment of part 1. Luckily, in the previous presentation, which I really liked, you understood what was part 1 and part 2. The burden will be distributed, and there will be, possibility to view all clinical trials-related information, which was not the case in the previous legislation. So, if we look at the timeline, after the application, the timelines will be adopted, the submission will take place, then diverging times are completely avoided, and there is an authorization of clinical trial in member state concerned with that trial. So if we look at the major key points of the regulation, we see that it will streamline the part 1 of assessment. When it comes to part 2, there will be a very defined timelines, mentioned a couple of times. And then finally, after the clinical trial is submitted via the single EU portal and database, we will have responsible reporting member state RMS to conduct part 1 assessment. Again, a lot of text. So, believe me, I'm in charge of medicine shortages primarily in our organization. So clinical trials are very exotic, but I'm not doing them at my hospital. So please interrupt if you think it's you know everything. So when I was reading these legislation documents, I saw that they will be related to compliance issues with Chapter 5, which is related to informed consent, then compliance with the labeling requirements, completeness and adequateness of the investigational brochure. Then, as outlined in Article 7 of the regulation, there will be a compliance with the requirements for informed consent, compliance of the arrangements for rewarding or compensating subjects in each clinical trial, and compliance of the arrangements for recruitment of subjects mentioned before. A lot of compliances, I'm not going to go through this. This is very, for me, it was very interesting, and for my colleagues, at least those who are receiving information about the trials, approval procedure should not take longer than 60 days. So basically, both assessment parts are conducted in accordance with the timelines. Then the sponsors are notified, and then the trial can commence at each member state concerned. This is just a graphical interpretation because we mentioned this a couple of times. It's going to be far more efficient now, because before the regulation, you'll see you have member state 1, member state 2, and member state 3. The same trial starts in different times in each country, and as mentioned in the previous presentation, you need to apply to every national competent authority and ethics committee. Now it's completely different. You have single submission and validation, and the trial starts in all member states concerned. There are 4 application types as before. So you have initial application, substantial modification application, and non-substantial modification application. And finally, if you want to add another country concerned into the trial, you can also apply via this portal. So applicants are assessed by appropriate regulators, and the assessment part 1 is carried by the reporting member state, while the assessment part 2 is carried by the member state concerned, which we already heard before. What's also very interesting, if you look about the regulation 070, it's not 007, we have 10 more, which is an EU regulation related to the publication of clinical data for medicinal products. There is a difference when you compare this policy and the clinical trial regulation. I'm not going to read the table, but the most important thing in 2020, we expect to have all clinical trial-related information generated during the life cycle of the trial, ready to be accessed by those who would like to see the information on a single place. You will not have to search for information, at least as I'm informed. So this is something which maybe is one of the most important issues compared to the previous legislation, and of course the application process that I mentioned. And finally, if you look at the draft, at least what I've seen from the single portal database, you have 3 separate places, one for sponsors, one for public, and one for authority. So sponsors will be able to easily start with all these applications that I mentioned, and the public will be able to access information they're interested in, and finally the authorities will have their own workplace. This all sounds very perfect and ideal. I'm afraid how it will work in spring 2019, but for now it looks very functional. It looks completely different than the directive. And the key benefits of the regulation, except for the fact that I mentioned this 300 times, it will be harmonized single portal admission process, which makes many of those involved in clinical trial application happy people. It will improve collaboration between the countries, and it will increase transparency of information in clinical trials, and finally, high standards of safety for all participants in EU clinical trials. I would also like to say that the reason why I'm so speaking quickly is the fact that I need to go back to my hospital because my general manager wants additional data about issues in our hospital pharmacy. So I would like to ask you if you have any questions, if you are particularly interested in EAHP, following our path as hospital pharmacists or clinical trials. And of course I want to say that we're very happy that we have Nate. I don't know if Nate is here. No, Nate is doing, no, you don't need to disturb him. Nate is an intern in our office in Brussels, and we are really, really happy that we continue our good collaboration with EPSA. Thank you very much. No questions.
Thank you very much one more time for your very nice lecture. So, as I told you before, I have some information before you can go to the coffee break. Remember that after the coffee break we will have one more session and we will proceed to the panel discussion. So prepare your questions and feel free to engage with our speakers. Remember also the competition of the educational outcomes that is occurring and started today. So feel free to submit your educational outcomes near the merge table. And speaking of this, during the coffee break we will have EPSA merchandise as well, so feel free to go there and check out what we brought for you. Also, there's the social media competition. All the information is being posted on Facebook, and remember the hashtag social media competition. And for now, that's it. Please proceed to the coffee break and see you soon.
A lot of things. Also, I need to pee.
Okay, I can
Isn't it possible to be up there?
Up there?
Because we'd be closer to the source of the sound.
I'll ask.
Maybe that's a good thing to start, and then I'll meet back again here in like 5.
Are you sure we're gonna have a good angle on video?
No, I'm not sure.
Yeah, I mean, it's gonna be from the up from first perspective, bird's eye perspective.
Okay, I'll just first tell Mihai that oh god, there's gonna be a line for the toilets. Okay, never mind.
How many people are watching?
5.
5. Oh.
Yeah.
We're on fire.
Oh yeah. It's been worse, as Mihai says. Oh, maybe I should stop live streaming first. Wait.